The spinal cord is the least developed in the neuraxis, following behind the development of the brain. It begins at the spinal medullary junction and runs in the vertebral canal to reach the coccyx. The position soon changes to the adult type by differential development of the two structures. The vertebral canal formed within the vertebral bodies continues the development while spinal cord lags behind. The result is that the spinal cord at birth ends at the level between L1 and 2 of the vertebral canal. The spinal cord then runs down from thence as filium terminale, which is just an extension of pia surrounding the central nervous system and intimately related to the brain and the spinal cord. The filium terminale extends to S2 in the vertebral canal and then becomes invested in dura mater. From this point, its extension to the coccyx is formed by the coccygeal ligament. It has the following morphological features: an expanded cervical portion, which is called the cervical enlargement. This is caused by the cervical and brachial plexuses. It also has that lumbar enlargement caused by lumbar and sacral plexuses. Its termination at the level between L1 and L2 of the vertebral canal forms a small protrusion called the conus medullaris. The spinal cord is invested in meninges, which is continuous with the meninges of the brain. They are 3 in number. The pia, which is the most internal and most intimately related to the spinal cord. The arachnoid, which is separated from the pia by a large space called the subarachnoid space. This space is accessible via lumbar puncture. In lumbar puncture, the doctor is interested in either diagnosis or treatment of cerebrospinal disease. Hence, he may wish to obtain cerebrospinal fluid for culture of foreign organisms or obtain the chemistry to determine its glucose or protein content, which may be suggestive of certain diseases. He may also be interested in injecting drugs into the subarachnoid space to treat conditions such as cerebrospinal infections. The commonest of these kind of infections is cerebrospinal meningitis. The subarachnoid space is expanded to the level of S2, where the filium terminale ends. It is more enlarged between the conus medullaris and S2, where the filium terminale ends. Hence, this position is used most extensively to assess the subarachnoid space for lumbar puncture in order to avoid the spinal cord. The dura mater is the most external covering of the brain and the spinal cord. In lumbar puncture, the procedure, which is a clinical one, involves the passing of hypodermic syringe with needle through the back into the skin between L2 and L3, or L3 or 4, or sometimes below L5. But L2 and 3 is used most extensively. The needle passes through the following layers: the skin, fascia, erector spinae tendons, posterior longitudinal ligament, intervertebral disc, anterior longitudinal ligament, ligamentum flavum, dura mater, arachnoid mater, and subarachnoid space. Epidural space. This space exists outside the meninges and specifically outside the dura mater. It is found in an expanded form below S2, where the filium terminale ends and the dura mater merges with S2. Below the level, the space is an expanded epidural space, which can be used for epidural anesthesia for operations on the perineum, and especially during parturition. The cauda equina represents the nerves which issue from the lower parts of the spinal cord, such as the sacral and coccygeal segments. They angulate downwards in order to escape from their corresponding intervertebral foramina. Hence, they form a column of nerves which surround the conus medullaris and accompany the filium terminale to reach the coccygeal ligament and S2 vertebra. These nerves are the ones easily blocked by either a lumbar puncture or an epidural anesthesia. It is equally important to mention that lumbar puncture is also used for anesthesia and is effective for abdominal and perineal procedures. Damage to this column of nerves, that is the cauda equina, may cause what is known as cauda equina syndrome, because they form a column which can be easily displaced by the needle. They do not constitute much danger to the spinal tap, that is another name for lumbar puncture, or anesthesia. The structure of the spinal cord. The spinal cord is made up of the following structures: the covering of meninges, white matter which follows the meninges, gray matter which follows the white matter, and the central canal. The gross structure of the spinal cord is as follows. The spinal cord begins at the spinomedullary junction, placed at the level of the foramen magnum. It then runs from this level to the junction between L1 and 2 vertebrae, where it actually ends as the conus medullaris. It is provided with lumbar and cervical enlargements, and also a slim portion at the thoracic level. Plexuses of spinal nerves are formed at the following levels: cervical plexus from C1 to 4, brachial plexus C5 to T1, lumbar plexus L1 to L4, sacral plexus L5 S1 to S4, and coccygeal plexus S5 and coccygeal 1. A spinal nerve exits from the spinal cord at its corresponding level of intervertebral foramen. It emerges initially as rootlets from two sides: dorsal rootlets, which are more than one, and ventral rootlets, also more than one. The rootlets come together to form the root of the nerve, which is just one, that is T1 or C5, etc. Motor nerves issue from ventral horn of the spinal cord, while sensory nerves issue from the dorsal horn. The sensory nerve appears at the dorsal aspect, while the motor nerve appears at the ventral aspect. Sensory nerves are mainly from the substantia gelatinosa. They form a dorsal root ganglion on the root nerve, at which their pseudo-unipolar neurons have their cell bodies. Note, there is no junction in the dorsal root ganglion except cell bodies of neurons, as opposed to the junctions that occur in autonomic ganglia. This occurs for both somatic and autonomic nerves, so long as they are sensory nerves. Their cell bodies are provided with two processes: a central process and a peripheral process. The central processes enter the spinal cord at the posterior horn, specifically at the substantia gelatinosa for somatic nerves, and the dorsal part of the intermediolateral horn for autonomic nerves. The peripheral processes are connected to interoceptors within the viscera for autonomic fibers, but connected to esteroceptors in the skin and the periphery for somatic nerves. Welcome to Spinal Cord Part 2. Motor nerves are derived from two main nuclear groups in the ventral horn of the spinal cord, the nucleus motorius lateralis and the nucleus motorius medialis. They are also divided into two types, that is, these nerves: somatic nerves, which approach the myoneural junctions or their motor end plates directly; autonomic nerves, which enter their corresponding autonomic ganglia initially as first-order neurons coming from the ventral part of the intermediolateral horn of the spinal cord. These first-order neurons are called preganglionic fibers. Second-order neurons then emerge from the ganglia and are then called postganglionic fibers. But in order to enter the spinal nerves, the autonomic nerve fibers make a different journey. The preganglionic fibers form the white rami communicantes. The singular is white ramus communicans. The postganglionic fibers then leave the autonomic ganglia via the gray rami communicantes. The singular is gray ramus communicans. The difference between the two is via their medulation. Gray rami are non-medulated, while the white rami are medulated. Internal organization of the spinal cord. The spinal cord has the following structures internally: central canal, gray matter, white matter, meninges, and other structures. This canal is formed from the primordial neural tube. It is lined by ependymal cells. It is continuous superiorly with the ventricles of the brain, which are also lined by the same cells. It is placed in the center of the spinal cord and contains cerebrospinal fluid. The gray matter of the spinal cord is gray because of the concentration of cell bodies of neurons in this matter. These form nuclei or nuclear groups from which axons issue and run into the horns of the gray matter in order to escape out of the spinal cord, that is, if they are motor neurons, or in order to enter the spinal cord via the horns if they are sensory neurons. The gray matter is also provided with the following horns: the ventral horn for the motor nuclei, which we had said before are nucleus motorius lateralis and nucleus motorius medialis. In the intermediate horn, it is divided into two: an intermediolateral horn running from T1 to L3, and an intermediomedial horn, which is of less importance as the intermediolateral horn. They contain nuclei of autonomic nervous system. The posterior horn of the spinal cord contains nuclei, which are second-order neurons of the sensory pathway. They are divided as follows. The most external is the nucleus posterior marginalis. Next to this is the substantia gelatinosa. Next to that is the column of Clark, or the Clark column, which extends from spinal cord segment C8 to L3. Rexed describe a lamination of the spinal cord, so that we now have 10 laminae or zones in the gray matter. Lamina 1 is the nucleus posterior marginalis. Lamina 2 is the substantia gelatinosa. Laminae 3 to 5 represents the column of Clark. Laminae 6 to 7 are placed between the central canal and the ventral horn. Laminae 8 to 9 are found around the ventral horn. Lamina 10 surrounds the central canal. Red Z lamination is used to describe the cytoarchitecture of the spinal cord and also used for comparative anatomical purposes. The white matter of the spinal cord is divided into three parts for the ease of description. We have the posterior column, which extends from the posterior septum to the posterolateral sulcus. You have the anterior column, which extends from the anteromedian fissure to the anterolateral sulcus. Then we have a lateral column, which extends from the anterolateral sulcus to the posterolateral sulcus. The posterior column has several tracts. The column is a funiculus, which is a collection of tracts. The white matter contains tracts which are both ascending and descending. The ascending tracts end in upper levels of the neuraxis, such as the medulla, the thalamus, and the cerebral cortex. The descending tracts come from the cerebral cortex and also from the extrapyramidal system. The posterior column has basically two fasciculi which are all ascending, the fasciculus cuneatus and the fasciculus gracilis. The grassa fasciculus is placed most internally between the posterior septum and the posterior intermediate sulcus. The fasciculus cuneatus joins the grassa fasciculus at higher centers of the spinal cord. They both carry primitive sensation from to the medulla. These sensations are the most primitive and are found in lower vertebrates, also associated with the medulla. They include pain, deep touch, pressure, vibration, stereognosis, and two-point discrimination. The extrapyramidal system associated with the spinal cord contains the following tracts: olivospinal tract, rubrospinal tract, vestibulospinal tract, and tectospinal tract. These are the major extrapyramidal descending tracts, although other tracts, which may not be as important, are available. Another tract worthy of note is the reticulospinal tract. This tract takes off from the reticular formation, which may be from the medulla pons or midbrain. The fibers descend into the spinal cord. The fibers act on the lower motor neuron, so that when the upper motor neuron is damaged, they provide a form of spastic response to the lower motor neuron. Ordinarily, they keep the action of the lower motor neuron in check, so that they do not overact or provide or balance for their action. Other tracts of the spinal cord are as follows: short ascending and descending tracts of the spinal cord, which are called the fasciculus proprius. The fasciculus proprius surrounds the gray matter of the spinal cord and is the first tract, usually after the establishment of a simple arc reflex to be established in the fetus. It provides quickening that the mother feels at about the 5th month of gestation, when the fetus begins to kick in the tummy. Tracts can be damaged by the following: injury to spinal cord or degenerative diseases. When it involves injury to spinal cord, it follows the following pattern. It can be either complete transection of the spinal cord at a particular level, for example C6. This is bilateral and may cause one of the following depending on its level. At a level at or above C2 or 3, it is not compatible with life. This is mainly due to the involvement of the phrenic nerves, which assist the movement of respiration as provided for by the diaphragm. At the level of C4, it may cause quadriplegia, which is paralysis of all the 4 limbs. At lower levels, it may cause paraplegia. This is because it spares the brachial plexus and affects the lumbosacral plexus, thereby paralyzing the lower limbs but sparing the upper limbs. The most easily topical of the injuries to the spinal cord is the so-called Sekot syndrome, which is the spinal cord hemisection. In this condition, the sensation is affected below the lesion on the opposite side, but motor function is affected on the same side. Also, there is rhomboedism on the same side. These are all related to the various tracts of the spinal cord, whether they are descending or ascending. There is also loss of sensation on the same side for the particular segment in which the damage occurs.